Hello friends, my name is Mohammad Imran. I am a third year MBBS student. So in this video, we are going to discuss about Pseudomonas aeruginosa. This is one of the most important bacteria from the bacteriology portion in microbiology MBBS exam. So let's start with the Pseudomonas. Pseudomonas is a gram-negative bacilli. It is non-fastidious, obligate aerobe, and it is motile. It is non-fermenter. It does not ferment any sugar but utilizes the sugar oxidatively. It is oxidase and catalase positive, and one of the characteristic feature of Pseudomonas is it is pigment producing. Now coming to the virulence factor and pathogenesis. Colonization. How does the Pseudomonas gets colonized on the host? By pili or fimbria or by polar flagellum. Now coming to the important part, that is toxin mediated immune evasion and tissue injury. There are two types of toxin. The first one is non-diffusible toxin. In non-diffusible toxin we have exotoxin, SUTY. You can remember this by SUTY. The second type of toxin is diffusible toxin. In diffusible toxin we have exotoxin A, proteases, phospholipases, hemolysins and pyocyanin. And the most important one in diffusible toxins is exotoxin A. It inhibits protein synthesis by inhibiting EF2, and EF2 is also inhibited by diphtheria toxin. Now coming to the third part of virulence factor, that is host inflammatory response. It is a defense mechanism, and host inflammatory response is seen against lipid A of lipopolysaccharide and flagellin. Now coming to the most important part, that is pigment production. There are four types of pigment. The first one is pyocyanin. It is a blue-green pigment and it is produced only by Pseudomonas aeruginosa, not by other species of Pseudomonas. Now coming to the second one, that is pyoverdin. It is greenish yellow pigment and it is produced by all species. The third one is pyorubin. It imparts red color, and the fourth one is pyomelanin. It imparts brown black color. So there are total four pigments. First one is pyocyanin, pyoverdin, pyorubin and pyomelanin. The fifth subtopic of virulence factor is alginate coat. The mucoid species of Pseudomonas produces a alginate coat around the bacteria, so this leads to the formation of biofilm. The biofilm formation helps in the adhesion to purulent mucus, and this leads to infection in cystic fibrosis patient. So we can see that in patients of cystic fibrosis, Pseudomonas infection is very common because of biofilm formation. The sixth point here is capsule. The capsule inhibits phagocytosis. Pseudomonas is multi-drug resistant, multi-disinfectant resistance, and the wide temperature range, it can survive between 5 to 45 degrees Celsius. Now coming to the clinical manifestations of Pseudomonas. So the Pseudomonas produces a variety of infections. So I have tried to compile everything in a single image, which would be very helpful during examination. So let's start with the eye. It produces corneal ulcers and endophthalmitis. In ear, it is responsible for swimmer's ear in children and malignant otitis externa in adults. In lungs, we are going to see pneumonia, ventilator associated pneumonia especially, and it also produces chronic respiratory tract infection in cystic fibrosis patient because of biofilm production and bronchiectasis patients. And Pseudomonas also produces Pseudomonas dermatitis. It is a skin infection. It also produces toe web infection. It is also responsible for meningitis in post-operative patients, and in heart we are going to see infective endocarditis. And the most important one here is it is responsible for burn wound infection. It also produces green nail syndrome and is also responsible for UTI, bone joint infection, osteomyelitis and cellulitis. The characteristic feature, blue-green pus, is seen in cellulitis by Pseudomonas, And the last one is ecthyma gangrenosum. It is a necrotizing condition seen in neutropenia and AIDS patient. So if you try to memorize this image, everything would be covered in the aspect of clinical manifestation of Pseudomonas. Now coming to the lab diagnosis of Pseudomonas, the first one is sample collection. Pus, wound swab, urine, etc. are collected. In direct smear, we are going to see gram-negative bacilli and pus cells. In gram staining, we are going to see gram-negative bacilli and pus cells. Now let's do the culture of Pseudomonas. The first one is peptone water. In peptone water, we can see uniform turbidity with surface pellicle formation. The second one is nutrient agar. In nutrient agar, we are going to see large, opaque, irregular colonies with metallic sheen. The important point here is it produces metallic sheen iridescence. And the second most important point here is we can see pigment production. As you can see, the green color in this image is because of pigment. And this pigment production can be enhanced by using another media known as King's media. Now coming to the blood agar. In blood agar, we are going to see beta hemolytic gray colonies. As we can clearly see in this image that there are gray colonies, beta hemolytic gray colonies. Now coming to the McConkey agar. In McConkey agar, we know that it is a non-fermenter. So it should be producing pale non-lactose fermenting colonies. And the selective media for Pseudomonas is cetrimide agar. You have to memorize cetrimide agar, that is a selective media for Pseudomonas. Now coming to the culture smear and motility testing. Again we are going to see gram-negative bacilli and motile. Motility testing, we are going to see it is a motile bacilli. The biochemical properties of Pseudomonas. As we already know that it is oxidase and catalase positive. It is a non-fermenter and produces non-lactose fermenting colonies in McConkey agar. In OF test, we are going to see oxidative pattern. In ICUT, indole test is negative, citrate test is positive, urease test is negative, and triple sugar iron test is also negative. So in ICUT, only citrate is positive and rest other tests are negative. In antimicrobial susceptibility testing is done for knowing the antimicrobial that is suitable for killing the Pseudomonas that is responsible for the infection. It is done on Mueller-Hinton agar by disc diffusion method. So ending this lecture on a positive note that this video is dedicated to my school friends Bhavya Pandey, Sarthak Sharma and Sadaf for helping me to restart my YouTube with their support and motivation.